I'm happy to present some data on ETX-155, which is a neuroactive steroid. It's not the first one to name. It is a positive allosteric modulator, so a PAM. And there are some differences here, and one of it is that the development strategy is to develop this for both mood disorders and epilepsy. The mechanism of action of the GABA PAMs is clinically validated with 2 approved compounds, both in depression and in epilepsy. And it is important to know that this compound also both targets the synaptic and the extrasynaptic receptors. We have observed robust anticonvulsant activity in preclinical models. We have limited the amount of models because of the available clinical validation of the mechanism, but in both the PTZ model and the MES model, we have seen those related improvements of efficacy similar to other compounds in the series of this mechanism. In addition, there is also robust anxiolytic and antidepressant activity in preclinical studies. There is efficacy in the elevated plus maze, so the animals don't like to be in the elevated position when it is not protected. And so there is increased time in these open arms of the maze. And in the forced swim test, where rats have to try to escape by swimming, there is reduced time of immobility, which is when they give up swimming, and more time climbing and swimming actually to try to get out. So these are very established models of anxiolytic and antidepressant activity. We have conducted a phase 1 study in healthy subjects last year. The study was conducted in Canada. And we were able to dose single doses from 5 to 200 milligram with a single ascending dose design with 6 active and 2 placebo subjects per cohort. The compound was generally well tolerated. In the 200 milligram arm, there were 2 sentinel subjects, which is now standard state of the art of doing just the 2 subjects initially. One of them wasn't active, the other one was on placebo. In that subject, developed severe adverse events of somnolence and ataxia. So we stopped dosing in the study and determined that the maximum tolerated dose, therefore, is 135 milligram. You can see that there is, you cannot actually see it, but there is dose proportional PK, and it's also linear increasing exposures with increasing dose. We then conducted a 7-day multiple dose study with 60 milligram once daily. One cohort received this in the morning, and we observed that there was somnolence in some subjects occasionally within the couple of hours post-dosing. We then repeated that with evening dosing and observed that there was much less somnolence. We also looked at next morning sleepiness, and there was no impact on awakening. People felt fine the next morning. So we think that it has advantages to dose this medication in the evening. We did not reach a steady state, and although the half-life seemed to be approximately 24 hours after single dose, the actual collection time was probably not enough, and that was confirmed by a second study where we dosed for 14 days, and steady state was achieved by day 8. You see the exposure curves here on day 1 and day 17. There were 3 days of non-dosing in between. That's why it's 17 and not 14. This was also well tolerated. That this was an evening dose. And the exposure after in steady state with 60 milligram is similar to what you see at single dose with 120 or 135 milligrams. So there is approximately a doubling of the exposures due to accumulation with this long half-life. The actual half-life is around 40 hours at steady state. The adverse events are listed here. So the main adverse event is somnolence. So actually similar to placebo in the 7-day. And in the repeat dose, there were six subjects with somnolence in active and 2 in the placebo, which is an expected side effect profile for this class. Importantly, we have observed that there is essentially no clinically meaningful food effect. There is a slight increase of the area under the curve, but no essential, maybe a slight decrease of the Cmax, which is different from some of the other compounds, where especially the SITCH compound has a much more marked food effect, so that you always have to take this with food. You see the difference between the fed state and the fasted state and the curves on the right. So there is essentially not much of a difference there. So in conclusion, we think this is a potentially clinically differentiated neuroactive steroid. And it is, we have very robust preclinical data. What I didn't show is that also evidence of target engagement with increased beta activity in rat EEGs. The compound was well tolerated in phase 1 studies, and the evening dose seems to be the preferred dose, and this has been better tolerated than the morning dose. The half-life is long, so it's suitable for once daily administration. We are currently conducting a photosensitivity model like many other people are doing. This study is underway. We have observed PK data from the first 3 subjects in this study, which was lower than it was in the previous phase 1 studies. And we are looking at the root cause of this. It was not a dosing error. It was not an assay error. It is an issue with the current batch of the drug product, which is different from the one that we used in the phase 1 studies. This has been a press release. I'm not telling you something that's not public, but so we are looking in the causes and hope to start our depression studies as soon as this has been resolved. And thank you for your attention.